All right, everyone. So today we're diving into Dr. Rosario's BIO259 lecture on muscle contraction.
Seriously, my brain is still trying to process all that.
Me too.
Right.
This deep dive is definitely needed.
For sure.
Like, I thought I knew how muscles worked, basic stuff.
Yeah.
But Dr. Rosario just blew my mind.
Right. It's way more complex than I ever imagined.
It's a little complex, sure, but also just keep in mind, as long as you have a couple of basic concepts down, it's actually not that bad. Like, this is just one of those topics that's a lot easier to understand than it is to memorize.
Totally. So to kick things off, can we talk about how muscle cells are basically in a league of their own? Dr. Rosario really emphasized that.
Yeah, I mean, they're basically super specialized for shortening.
So not your average skin cell, huh?
Exactly. See, most cells are filled with this fluid, cytosol, which makes up most of their volume.
And while muscle cells also have some cytosol, they're like really stuffed full of structures that are super important for muscle contraction.
They're packed with those myofibrils, right? The ones Dr. Rosario kept mentioning.
You got it. Myofibrils are like long protein threads that take up the lion's share of a muscle cell space.
And those are the things that do the heavy lifting, so to speak.
Precisely.
Yeah.
They're the key players in muscle contraction.
And the reason that they're so important is because they contain all of those myofilaments that I mentioned in class.
That's so cool. So it's like our muscles are these little factories, and the myofibrils are the machines working overtime. But Dr. Rosario, didn't you say those myofibrils are made up of even smaller threads, the myofilaments?
Yeah, those are what we usually refer to as the thick and thin filaments, and it's the interaction of regions of the thick filaments grabbing onto the thin filaments and pulling.
Oh, so it's like a microscopic tug of war happening inside our muscles.
Yeah, and we're going to dive deep into that tug of war mechanism.
But before we get to that, can we back up a sec and talk about the sarcoplasmic reticulum?
Sure.
I always stumble over that word.
Oh, tell me about it. It's a mouthful.
Yeah, that's why sometimes I just refer to it as the SR. That's good enough.
The sarcoplasmic reticulum is like a specialized storage network for calcium ions.
Calcium. Why does that sound familiar?
Because, as you'll soon see, calcium is absolutely essential for making those muscles contract. It's like the signal that sets everything in motion.
That's right. But I usually like to think of it as like a light switch, right? Like having calcium present is like the switch being turned on. Removing calcium is like having the switch turned off.
Okay, so it's like the calcium's the signal, and the myofibrils are just waiting for the green light to start contracting.
This is already making so much more sense. All right, so we've got these jacked up muscle cells with their myofibrils primed and ready.
And they're just waiting for that calcium signal.
But how do they actually contract? What's happening at the microscopic level? Dr. Rosario, I think we're ready to delve into the world of sarcomeres.
Okay, Dr. Rosario, so we're zoomed in on these sarcomeres, the little units of muscle contraction. What are we looking at here?
Okay, so let's just remind ourselves what sarcomeres are. They are the basic unit of skeletal muscle, and they're made up of a bunch of myofilaments that interact with each other, right? So right in the middle of the sarcomere, you have the thick filament with multiple myosin heads, and those myosin heads are able to stretch out and grab. What do they grab? Well, they grab the thin filaments, right? And finally, the thin filaments are really strongly attached and anchored to these things that we call Z-discs. So to answer your question, what is a sarcomere? It's all of the filaments, the thick and thin filaments that we find in between two specific Z-discs.
Okay, that makes sense. But Dr. Rosario, what are these filaments made of?
Yeah, what's the deal with these thick and thin filaments?
Well, let's just start with the thick filaments first. They're mostly made up of a protein called myosin.
Myosin, huh? I vaguely remember that from bio.
And here's where it gets even more interesting. Each myosin molecule, picture it like a golf club. Long handle, two heads.
Wait, two heads, like a two-headed golf club. And those two heads have a very important job. They bind to another protein. This one's called actin.
Actin.
And guess what? Actin is the main protein making up those thin filaments.
So it's the myosin heads grabbing onto the actin, like a microscopic handshake.
You got it. And that, my friends, is what we call a cross bridge. This little connection between myosin and actin, that's the foundation of muscle contraction.
Wow. So all our movements, everything comes down to these tiny little handshakes.
It's pretty mind-blowing, but it's not just a simple handshake. It's more like a carefully choreographed dance.
A dance. Okay, now you have to explain this.
It's called the cross-bridge cycle, and that's how those myosin heads actually pull those Z-lines closer together, making the muscle contract.
Okay, Dr. Rosario, you've got to walk us through this cross-bridge cycle. This is getting good.
All right, Dr. Rosario, spill the tea. What is this cross-bridge cycle all about?
All right, so the cross-bridge cycle, It's basically the mechanism that we use to continually form and break cross bridges. So remember, once the myosin heads are stretched out, they're essentially ready to form a cross bridge.
Ready to make that handshake right. Exactly. But there's a little catch.
Uh-oh. What's the catch?
The binding sites on actin, they're kind of blocked.
Blocked? By what?
By another protein, actually. It's called tropomyosin.
Tropomyosin. So it's like a bodyguard for actin.
I like that. It's like tropomyosin is saying, hold on there, buddy. Not so fast.
So how does myosin ever link up with actin then?
That's where our friend calcium comes in.
Calcium again.
Remember how we talked about the sarcoplasmic reticulum storing calcium?
Right, right.
Well, when calcium gets the signal, it floods the muscle cell and binds to another protein called troponin.
Troponin, what does that do?
So troponin is bound to the tropomyosin that's blocking the cross bridge formation. And when calcium binds to troponin, it actually causes the tropomyosin to be pulled slightly out of the way.
So the bodyguard steps aside.
Precisely. Now that myosin binding site on actin is wide open.
And myosin can finally make its move.
Fueled by ATP, right?
Exactly. ATP gives that myosin head the energy to bind to actin, forming that cross bridge.
Okay, so we got the handshake. But then what? How does the muscle actually shorten?
That's the beauty of this whole cycle. The binding of myosin to actin, it triggers this thing called the power stroke.
Power stroke. Sounds intense.
It is. Basically, the myosin head uses the energy from ATP to swivel, like a lever, pulling that actin filament towards the center of the sarcomere.
So it's like myosin is reeling in actin, bringing those Z lines closer together.
Perfect analogy.
Wow. So each tiny myosin head is pulling like crazy, but they can't just stay attached, right?
You're right. Another ATP molecule comes in, binds to the myosin head, and that causes it to detach from actin.
So they need ATP to let go too.
Exactly. ATP is essential for both the attachment and detachment phases of this cycle.
It's like ATP is the oil that keeps this whole machine running.
I like that. It's the energy currency that makes everything happen.
Yeah, the ATP is really important for energy, but let's just keep this little detail in mind. When ATP binds to myosin to break the cross bridge, we don't actually have to spend any of that ATP's energy to do so, right? As long as the ATP attaches, the cross bridge will break.
So the cycle repeats. Myosin grabs, pulls, releases, grabs again, pulls again, and so on.
You got it. And this cycle is happening incredibly fast across millions of sarcomeres to produce even the smallest muscle contraction.
That's incredible. And all this is going on inside our muscles every time we move.
Every time.
I'm never going to look at my muscles the same way again.
Me neither. And to think, this all started with those lectures in BIO259.
Right. Dr. Rosario, thanks for breaking down such a complex process. This deep dive has been amazing.
You know, I am always happy to break down some of these complex ideas into smaller, understandable parts. So thank you so much for having me on here. Thank you for being great hosts and for asking me some really great questions.
My pleasure. Always happy to talk about the wonders of muscle contraction.
Until next time, everyone, keep those sarcomeres cycling.